Welcome back to the OpenHPI course Code of Life. Today we are talking about compiling the code from the cell core, and this is important because it's referred to transcription and translation. It's a complex process. First of all, we need to step one step back to understand how does the product look like. Therefore, I'd like to introduce to you the so-called amino acid coding sun. This is a structure you see here. It's typically read from the center to the outer segments, and you have three kinds of cycles. And you see the different nucleobases here. You see the A, C, G, and U because we are talking about the RNA level. And in the first ring, you have like 4 different kinds of alternative. In the second, you have again 4 different alternatives, and in the last one also. That means you would expect 4 multiplied by 4 multiplied by 4 alternative amino acids. But in the end, in the human body, you only find 20 so-called canonical amino acids. That means you have here some kind of redundancy to protect already and to increase the robustness of the products of the DNA. So you see there are some letters at the outer circle which are referring to the actual name of the amino acid, which is not so important in our context here. However, you see also very specific signs here. For instance, the start codon here down, it's ATG. ATG is for every kind of ribosome, the information here you have to start decoding. And then you see these 3 dots up there, these 3 dots referring to stop codons, right? And these are important to stop the protein synthesis. So what you take home here is very important. You see there is already robustness and integrated by redundancy in the code. So let's switch to the first step. It's called transcription. Transcription is the process of copying DNA from the cell core. So to get the source code out of the cell core into RNA, which is then transported to the cytoplasm. That means the information stored on the DNA in a protected area is replicated into the RNA, and then it's moved to the cytoplasm. And therefore, the RNA can be separated in 3 different kinds of purposes, so to say. The first kind of RNA is ribosomal RNA, or rRNA. rRNA contains the source code for building ribosomes. Ribosomes are our compiler. Recap this. So this is the source code for the compiler. The second version is the transfer RNA. Transfer RNA is important for getting the right amino acids which are flowing around in the cytoplasm, getting them, binding them, and bringing them to the ribosomes for further processing. And most probably the most important kind of RNA is the messenger RNA, because this exports the kind of source code which should be assembled by the ribosome. So it's the source code for the compiler to render some kind of objects here. In this case, it's the proteins which should be created out of that. Therefore, transcription is the first step to get the information out of the cell core into the cytoplasm. The next thing we need to talk about is the so-called translation. Again, you see here on the right-hand side, this is a ribosome, and the ribosome has the task to produce amino acid sequences, so-called proteins. Translation is the process of exactly creating these proteins using a template, and the template is typically the messenger RNA. That means out of the messenger RNA, you will form the right sequence of amino acids. And therefore, we have 3 phases. The first one is again the initiation phase. That means the ribosome gets some messenger RNA and detects the start codon. You can remember in the coding sun there's only one start codon, ATG. And this start codon, once it's detected, it knows now I need to find the right transport RNA components to bind to this kind of codon. This is done in the elongation phase. That means amino acids are bind to this transport RNA and then move to the ribosome, and the right one binds to the corresponding RNA strand. That means you see here on the top that these bubbles which are getting longer and longer, this is the amino acid sequence. Needs to be compiled by the ribosome. And last but not least, when the ribosome detects a termination codon or a stop codon, it stops its work and releases the product, the protein, into the cytoplasm. That means it stops compiling anything. So therefore, you know it's also important to have these start and stop codons at the right region. Once they are somehow affected, they can lead to completely different products. And how to even increase the variability using the same DNA. What you see here is the so-called alternative DNA splicing. On the top level, you see the DNA, how it is winded in the cell core. Then you see the RNA, which is read from the DNA, which is more or less the same blueprint. However, now with the same RNA, you can create different kinds of messenger RNA. On the left-hand side, you see, again, these 5 exons you see on top of that. You see exon 1 to 5 in different colors, and the first messenger RNA contains all of these 5. This is a 1-to-1 copy. The second one, you see, it does not contain the 3rd exon. And the third one even don't contain the 4th exon. That means even if you have the same RNA, you can use splicing to have certain kinds of adaptation in the products. And you see down here in the proteins, they slightly differ. These are the red highlighted regions. That means RNA splice or DNA splicing results in different messenger RNAs, so the ribosomes create different products, even if you have the same source code. That means you have an adaptation point, like a parameterization of the source code by using this slicing variant. So what to take home? You should think about the different kinds of how products, especially proteins, can be built from the DNA code from within the cell, that there needs to be transcription and translation involved to get this done. And next time we want to investigate what happens if something is going wrong here. Especially if some of the information which is in the DNA is somehow changed, altered, or even on the way to the ribosome something happens, and what could be the effects out of that. Therefore, stay tuned, and I'm looking forward to see you next time.